Have you ever noticed how some days you just, feel in sync, waking up refreshed, energy levels steady, and then other days it's like your internal calendar is completely off, leaving you groggy or maybe wired at the wrong times? We often blame our sleep, maybe a bad night's rest, but there's a powerful, often invisible conductor orchestrating these daily rhythms, your circadian clock. Today, though, we're not just talking about sleep. We're diving deep into something, well, far more fundamental for male health: testosterone. This deep dive, using insights from the latest research in the Asian Journal of Andrology, is going to reveal how these tiny internal timekeepers are intimately linked to male hormones and really overall well-being.
And this connection is truly remarkable. Testosterone isn't just a hormone for men. It's one of the most vital sex hormones, absolutely essential for, well, pretty much everything from reproductive system development and function to bone density, mood, overall vitality. The list goes on.
Right. It's kind of a big deal.
It really is. And what's fascinating here, what the research really digs into, is that its secretion isn't constant. It's not just a steady drip. It follows distinct circadian rhythms, meaning it predictably rises and falls over a 24-hour period. And this rhythm, it's directly influenced by your circadian clock genes.
Ah, okay. So the genes are calling the shots on the timing.
In a very real way, yes. So, throughout our conversation today, we'll explore the central mechanisms, like from the brain and the peripheral ways this regulation happens right down in the tissues. And then we'll look at the surprising impact these genes have on common male health issues. Things like late-onset hypogonadism, erectile dysfunction, male infertility, and even prostate cancer.
Okay, let's unpack this idea of the circadian clock first, just to make sure we're all on the same page.
Yeah.
Think of it as your body's master internal timer, right? Like a biological conductor leading this whole symphony of physiological and behavioral rhythms.
Exactly. A conductor is a great analogy.
It coordinates everything: hormone release, body temperature fluctuations, that crucial sleep-wake cycle, and even how individual cells manage their daily tasks, their own little cycles. And the stars of this show, you mentioned them, are the circadian clock genes. These specific genes, both central in the brain and also spread throughout the body, they're the ones directly regulating these essential 24-hour rhythms.
Both central and peripheral. That's key.
And here's where it gets, I think, really eye-opening for a lot of people. When disruptions hit this delicate system, things we encounter all the time, like working night shifts, maybe too much late-night screen time, or even what scientists call social jet lag.
Right, that inconsistency between your weekday and weekend sleep schedules.
Exactly. When those things happen, it can throw these genes off balance, and a misaligned clock, it turns out, can directly lead to abnormal testosterone levels.
Precisely. And the master conductor stand, this sort of central control site, is a tiny region deep in your brain called the suprachiasmatic nucleus, or SCN. It sits in the hypothalamus.
SCN.
Yeah, SCN. It's incredibly smart, biologically speaking. It directly receives light information straight from your eyes. That's the primary cue. And it uses that light signal to set your body's precise 24-hour cycle. Then, from the SCN, signals radiate out to orchestrate rhythms throughout the entire body.
So it's getting the time of day info from light.
Primarily, yes. But it's not just a top-down command structure. These same clock genes, they're also widely expressed in nearly every cell and organ. They form what we call peripheral clocks. And critically for our discussion today, these peripheral clocks are very active, very present in the testes.
Okay, so local clocks, right where the testosterone is made.
Exactly. The complete system is this beautiful interplay between the central SCN and these local peripheral clocks working together.
So we have the central maestro in the SCN, and then these local timekeepers in organs like the testes. But how do they actually keep time at a molecular level? What's the dance happening inside the cells?
Yeah.
It's a complex dance, but it's elegant. At its core, you can sort of imagine a biological dimmer switch operating on a very precise 24-hour loop. You have two key genes, CLOCK and BML1. Think of them as partners. When they team up, they essentially turn up the volume on a whole host of other genes, including two really important ones called PER and CRY. Now, as more PER and CRY proteins get produced, they build up and act as a brake. They basically go back and tell CLOCK and BL1 to dim the lights again to slow down production.
Yeah, feedback loop.
Exactly, a negative feedback loop. This constant ebb and flow, this turning on and turning off, creates that perfect, self-sustaining 24-hour rhythm. There are also some additional fine-tuning elements, other genes like NR1D12 and RORA that help keep the whole system incredibly precise. But that core loop, KLOCK-BMEL1 activating periphery and periphery inhibiting KLOCBMEL1, that's the engine.
That's a great analogy. A cellular dimmer switch for our internal rhythm. Very clear. And you mentioned these clock genes are definitely in the testes. But is it like uniform across all the cells there, or are some cells more important than others in this context?
That's a really important nuance, and actually it was a point of some scientific debate for a while. Some early studies looking at the whole testicular tissue maybe didn't show a super strong overall circadian rhythm in clock gene expression, and that could be due to differences between species studied or just the sheer complexity. There are many different cell types in the testes.
Right, makes sense.
But when we zoom in, the picture becomes much clearer. What's now considered unequivocal and truly vital for this discussion is that the Leydig cells.
The testosterone factories.
Exactly, the primary dedicated factories for testosterone production. Those cells absolutely express these clock genes with significant circadian rhythmicity. We're talking about genes like BMEL1, PER1, PER2, PER3, CRY1, CRY2, and NR1D1, all marching to a clear 24-hour beat within those specific testosterone-producing cells.
Okay, that's a critical distinction. So even if the whole tissue average is messy, the key players, the Leydig cells, are definitely on the clock.
Precisely. And while other cells, like the spermatogenic cells involved in sperm production, also have clock genes, their expression might not always show that same strong circadian rhythm. So this cell-specific activity within the Leydig cells is a real key takeaway here.
That distinction is crucial. It really narrows down where the critical action is happening for testosterone regulation. And it brings us directly to testosterone itself. You said it's not just a steady flow. Its secretion kind of dances to rhythms, not just daily, but even across a whole life cycle and annually.
That's right. There are longer-term rhythms, too, annual variations. But the circadian rhythm is perhaps the most impactful for our day-to-day physiology. It peaks typically in the morning hours for most men and then gradually declines throughout the day and evening.
Right. Which explains why maybe energy levels can feel different morning versus evening. So this raises a crucial question about aging. What does this rhythmic life of testosterone mean for health as men get older?
Yeah, it means quite a lot, unfortunately. Research consistently shows that in healthy elderly men, that strong daily circadian rhythm of testosterone often significantly weakens. It might become less pronounced, or in some cases, it can even become completely absent compared to younger men.
So the rhythm flattens out.
It often flattens out, yes. And it's not just the rhythm that falters. The Leydig cells themselves, those testosterone factories, also tend to become less responsive to the stimulating hormones they need as we age. Hormones like LH.
Ah, okay. So it's a double hit then. The timing mechanism gets disrupted, and the factory itself starts slowing down or not listening as well to the signals.
That's a good way to put it, a double hit. And understanding how this regulation works, both centrally and peripherally, helps explain why it can falter.
Okay, let's get into that. How does the central command, the SCN, actually control this rhythm?
Right. So the SCN, our master clock in the brain, takes that light information and converts it into endocrine signals, hormone signals that are eventually sent down the line to the Leydig cells. Two really key hormones in this pathway are melatonin and luteinizing hormone, or LH.
Melatonin, the sleep hormone?
Often called the sleep hormone, yes, but it does much more. It's secreted by the pineal gland, also under the SCN's control, and its levels naturally peak at night, like a chemical signal for darkness. Melatonin acts on the hypothalamus, another brain region, to inhibit the release of something called gonadotropin-releasing hormone, or GnRH.
Okay, so melatonin puts the brakes on GnRH at night.
Exactly. And GnRH normally tells the pituitary gland to release LH, and LH is the hormone that directly travels to the Leydig cells and tells them, okay, make testosterone. So when melatonin is high at night, it dampens this whole cascade. Less GnRH, less LH, and consequently less testosterone production. This is a big reason why LH and testosterone levels often rise and fall together synchronously throughout the day, peaking in the morning when melatonin is low.
Okay, that makes sense. Melatonin acts as a sort of nocturnal brake on the whole production line, signaling the body it's nighttime, time to power down testosterone synthesis. But does melatonin have a more direct role too? Does it talk straight to the Leydig cells?
It absolutely does. It's not just acting upstream in the brain. Melatonin also has a direct line to the Leydig cells themselves. It binds to a specific receptor right there on the Leydig cell surface called melatonin receptor 1. Binding to this receptor affects the expression of other things inside the cell, including a protein called CRH, which then inhibits another crucial protein called STAR.
STAR, what does STAR do?
Think of STAR as the absolutely essential shuttle bus. It's responsible for transporting the raw material for testosterone cholesterol from the outer part of the cell's mitochondria to the inner machinery where testosterone synthesis actually happens. No STAR, no cholesterol transport, no testosterone. So, by affecting STAR activity via CRH, melatonin can directly inhibit testosterone synthesis right within the Leydig cell.
Yeah.
And the proof is pretty strong. Studies in mice where they knocked out the gene for that melatonin receptor 1 showed significantly disrupted testosterone rhythms. It really highlights melatonin's direct importance at the local level.
Fascinating. And then, presumably, when testosterone levels rise, there's a feedback signal.
Yes, exactly. Once testosterone levels get high enough, they complete a classic negative feedback loop. The testosterone itself signals back to the brain, to the hypothalamus and pituitary, telling them to inhibit GnRH and LH release. It keeps the whole system in balance, preventing levels from getting too high.
Okay, so that covers the central control, the brain's role with SCN, melatonin, GnRH, LH, very intricate. But you mentioned peripheral regulation in the Leydig cells themselves. How do those local clock genes directly influence testosterone production beyond just responding to melatonin?
This is where those Leydig cells become really fascinating autonomous units in a way. Genomic research, looking at gene activity across 24 hours, tells us that maybe around 1/3 of all genes expressed in a cell change their activity levels with a circadian rhythm.
A third. That's huge.
It's a significant portion. And many of these rhythmically expressed genes are directly controlled by those core clock genes we talked about, like BMAL1 and clock. These two act like master switches within the Leydig cell, directly influencing the speed and efficiency of the crucial steps where testosterone is actually built. They do this by regulating the production of key enzymes, those specialized proteins that drive the testosterone production line forward.
Enzymes like?
For instance, BMAL1 and CLOCK directly control the genes for enzymes like STAR, that critical cholesterol shuttle bus we just mentioned, and another one called CYP11A1, which performs the very first chemical conversion step, turning cholesterol into pregnenolone, an early precursor to testosterone. Also HSD3 and HSD17, which are involved later in the pathway.
So they're literally controlling the production machinery. Is there concrete evidence showing this link? Like if you tweak the clock genes, you see changes in these enzymes.
Yes, absolutely. Very clear evidence from experimental studies. For example, if you genetically engineer cells or mice to overexpress BMAL1 to make more of it, you see a corresponding increase in STAR protein and other key enzymes like HSD17. Conversely, if you create knockout mice that completely lack a functional BMAL1 gene, they have significantly reduced testosterone levels. And when you look inside their Leydig cells, you find lower levels of STAR and CYP11A1.
Wow, okay. Direct cause and effect.
It strongly suggests it. And it's not just BMAL1. Similar findings exist for other clock genes. Mice with specific disruptions to PIR1 and PIR2, those brake genes, show downregulated levels of key testosterone-synthesizing enzymes like CYP11A1 and HSD3B, and consequently lower serum testosterone. Even NR1D1, part of that secondary feedback loop, has been shown to directly regulate other steroid synthesis genes. So the bottom line is these clock genes aren't just passive bystanders keeping time, they are actively dictating the pace and capacity of testosterone production minute by minute, hour by hour.
That makes perfect sense. Direct genetic control over the production line enzymes. But you hinted earlier there might be other, maybe more subtle, indirect ways these peripheral clock genes influence testosterone, beyond just managing those enzymes directly.
They absolutely do. It's not just about controlling the enzyme levels. They also influence the overall cellular environment in ways that indirectly impact testosterone synthesis. One major factor is oxidative stress.
Oxidative stress, right? Like cellular wear and tear.
Sort of, yeah. The internal redox state, the balance between oxidizing and reducing molecules, naturally changes within cells according to the circadian rhythm. Sometimes this leads to an increased production of reactive oxygen species, or ROS. You can think of ROS as like cellular exhaust fumes. A little bit is normal, but too much can be damaging. Leydig cells happen to be quite vulnerable to ROS damage.
And how does that affect testosterone?
High levels of ROS can actually inhibit testosterone synthesis. One way they do this is by reducing the expression of STAR, slowing down that critical cholesterol shuttle bus. But here's the crucial link back to the clock. Key clock genes like BMAL1 and PIR2 and even the hormone melatonin have been shown to have antioxidant properties. They can help inhibit oxidative stress by reducing ROS levels.
Ah, so the clock genes help protect the Leydig cells from their own metabolic byproducts, creating a better environment for making testosterone.
Exactly. They help maintain a healthier, more optimal internal environment. And another fascinating indirect route is through a process called autophagy.
Ontology, that's the cell's recycling system, right?
Precisely. It's the cell's natural recycling and cleanup crew. It's a process where cells break down and recycle old or damaged components, like worn-out mitochondria. And intriguingly, the number of these cellular recycling units, called autophagosomes, also shows a clear daily rhythm, suggesting a direct link to the circadian clock. For example, studies have found that mice lacking the BMAL1 gene have reduced levels of autophagy-related genes and fewer autophagosomes. They're not cleaning up as efficiently. Now, in Leydig cells, this recycling process is really important for two key reasons. First, it removes dysfunctional mitochondria, which are a major source of those damaging ROS we just talked about.
Right, reducing oxidative stress.
Exactly. And second, autophagy also seems to help promote cholesterol uptake into the cell, providing more of the raw material needed for testosterone synthesis. So by influencing the level of autophagy, clock genes can indirectly regulate testosterone synthesis by ensuring the cell's internal environment is clean, efficient, and well supplied.
It's truly incredible to think how all these tiny gears, from the genetic switches controlling enzymes to managing oxidative stress to cellular recycling, are all working together or, potentially not working together if the clock is off. So this brings us to the really crucial question. What happens when the clock goes awry? What are the real-world health implications when these delicate rhythms get disrupted? We're talking about pretty significant impacts, aren't we?
We really are. The real-world consequences are profound and becoming increasingly recognized. Things like shift work, which, as you know, affects a huge portion of the global workforce, maybe 10% to 30% of workers.
That's a staggering number.
It is. That, along with just chronic insufficient sleep or even subtle genetic variations in our individual clock genes, are now being clearly linked to a whole variety of male health issues. Studies, for example, consistently show that shift work can decrease total testosterone levels and even delay the timing of its peak production during the day. It essentially scrambles that carefully choreographed rhythm we've been discussing.
Right, so where do we see the biggest impacts? What specific conditions are linked?
Okay, let's start with late-onset hypogonadism, or LOH. This is basically the age-related decline in testosterone, often called andropause. While aging is the main driver due to declining Leydig cell function, circadian disruption seems to exacerbate it. Clinical studies paint a pretty clear picture. Non-standard shift workers, especially those who also suffer from sleep disorders, tend to have lower testosterone levels and report more severe LOH symptoms, things like low energy, mood changes, reduced libido.
So the lifestyle factor makes the age-related decline worse.
It certainly appears to. And as we touched upon earlier, that natural circadian rhythm of testosterone is often significantly weakened or even completely absent in many elderly men, particularly those diagnosed with LOH. The potential good news here is on the therapeutic side. In animal models, researchers have found that boosting the activity of certain clock genes like BMAL1, NR1D1, PER2, and CRY1, along with key steroid synthesis genes, actually shows promise in increasing testosterone levels. It suggests targeting the clock might be a future strategy for LOH.
Interesting. Okay, what about erectile dysfunction, or ED? Is there a clock connection there too?
There is, yes. Disruptions to our fundamental sleep-wake cycle are strongly linked to impaired metabolic health in general, and ED specifically seems to be part of that picture. We see, for instance, that night shift workers consistently report significantly lower scores on the IIF, the International Index of Erectile Function, which is a standard measure for assessing erectile health.
And what's the mechanism there? Is it just low testosterone causing ED?
Low testosterone can certainly contribute, but the clock's influence seems broader. A key mechanism for achieving an erection involves the nitric oxide, or NO, and cyclic guanosine monophosphate, CGMP, signaling pathway. This is the crucial system that allows blood vessels in the penis to relax and fill with blood. Intriguingly, this NO-CGMP pathway appears to be regulated, at least in part, by clock genes. For example, studies have shown that a mutation in the PER2 gene can lead to reduced NO production, and BMAL1 knockout mice exhibit signs of endothelial dysfunction, meaning their blood vessels don't function properly.
Wow. So it's not just about the hormone levels directly, but even the subtle cellular mechanisms of blood flow control. That's a critical insight. What about male fertility? You mentioned spermogenic cells also have clock genes.
Yes, male infertility is another major area where the circadian clock plays a significant role. It's fascinating, actually. We even see subtle seasonal changes in human semen quality reported in some large studies, with things like higher sperm count and concentration typically observed in the spring, and maybe more normally shaped sperm in the summer. Suggests a link to natural light cycles.
Like an annual rhythm for fertility.
Potentially, yeah. But artificial disruptions are a much clearer concern. Several studies have linked night shift work to an increased risk of infertility in some populations. The risk was nearly 4 times higher for shift workers. And direct experiments in mice really drive this home. If you simply reverse their light-dark cycle, mimicking chronic jet lag or shift work, it leads to a significant reduction in their sperm count.
Okay, so the disruption itself harms sperm production. How do the clock genes fit in?
Again, specific clock genes seem critical. Knocking out PER1 and PO2 in mice leads to downregulated expression of genes essential for spermatogenesis, the process of sperm creation, resulting in reduced sperm function. A specific mutation in the clock gene itself has been shown to reduce the ability of sperm to fertilize an egg. And knocking out CRY1 seems to increase apoptosis, or programmed cell death, among spermatogenic cells, leading directly to a lower sperm count. So multiple clock genes are implicated in maintaining healthy sperm production.
This is quite a list. LOH, ED, infertility. What about cancer? Does the clock influence prostate cancer, PCA, risk, or progression?
This is a really important and active area of research. And yes, circadian rhythm disorders are increasingly recognized as being closely related to prostate cancer occurrence and potentially its progression. There are genetic links appearing. Certain natural variations or polymorphisms in clock genes like BIML1, PER1, and PER3 have been associated with an increased risk of developing PCA in some population studies. And looking at tumor tissue itself, abnormal expression levels of CRY2 and BMAL1 have been linked to how aggressively the prostate cancer behaves its progression.
And how might that work at a cellular level? How do clock genes influence cancer cells?
Well, clock genes normally play a vital role in regulating fundamental cellular processes like cell division, proliferation, programmed cell death, apoptosis, and the cell's ability to respond to and repair DNA damage. These are all processes that go haywire in cancer. So it makes sense that disrupting the clock could disrupt these controls. For example, experimentally increasing the expression of PER1 in prostate cancer cell lines has been shown to inhibit their growth and actually promote apoptosis, encouraging them to die off. And conversely, there are now compounds being developed called NR1D1 agonists, which activate another clock protein, and these have shown potential in lab studies to suppress prostate cancer cell growth and their ability to migrate or spread.
This is truly eye-opening. It really connects the dots from our daily rhythms, something we might take for granted, to such a wide spectrum of really significant male health challenges. But these insights aren't just for, understanding the problems. They must open up some exciting new avenues for treatment, right?
Absolutely. And one of the most promising, I think, is the concept of chronotherapy. What's truly compelling here is the idea that it's not just about what medicine you take, but when you take it.
Timing is everything.
Timing is potentially crucial. Chronotherapy is all about strategically timing drug administration to sync up with the body's natural circadian rhythms. The goal is to maximize the drug's effectiveness while minimizing side effects on healthy tissues. For LOH, for example, we mentioned boosting clock genes might be a future approach. But even with current treatments like testosterone replacement, timing matters. Some studies suggest that administering low-dose testosterone ointment, like a product called Glomin, in the morning does a better job of mimicking the body's natural daily rise and fall of testosterone. This might improve symptoms more effectively and potentially reduce costs or side effects compared to less physiological dosing schedules.
That makes intuitive sense. Work with the body's rhythm, not against it. What about for prostate cancer?
For PCA, the potential is perhaps even more significant. Cancer cells often have disrupted clocks, but surrounding healthy tissues usually maintain their normal rhythms. Chronotherapy aims to exploit this difference. By timing the administration of anti-cancer drugs to specific points in the day when the cancer cells might be most vulnerable, example during DNA replication, and normal cells are relatively quiescent or resistant, you could potentially enhance the tumor cell killing effect while significantly reducing harm and toxicity to the patient's normal tissues. It's like hitting the target when its defenses are predicted to be down.
That's a really powerful idea. Turning our body's own internal rhythm into a therapeutic advantage is quite elegant. What about non-drug approaches? Are there practical things, lifestyle things we can do based on this knowledge?
Definitely. And these are often more accessible. Beyond medications, there are powerful non-pharmacological approaches that leverage our understanding of circadian biology. For shift workers, simply finding ways to adjust working hours, even slightly, or implementing strategies to stabilize sleep schedules on days off, can help mitigate the constant circadian disruption and its associated health consequences. It's challenging, but important.
Right. Easier said than done sometimes, but crucial.
Absolutely. Then there's phototherapy or light management. This could involve using bright light therapy in the morning to help reset the clock, or maybe more practically for many, using filters or special glasses to block shortwave light, particularly blue light in the evening and at night. This reduces the disruptive signal that artificial light sends to your SCN, lessening the interference with melatonin production and sleep.
So managing our light exposure is key.
Very key. And even dietary patterns matter. There's growing evidence for things like intermittent fasting or time-restricted eating. Sticking to a consistent eating window each day seems to positively regulate the circadian rhythms, not just in our main organs, but also in our gut microbiome, the bacteria living in our gut. And a healthy gut rhythm is increasingly linked to preventing obesity and metabolic syndrome, conditions which themselves are closely tied to many of the male health issues we've discussed, like LOH and ED. All these approaches point towards a future where consciously managing our internal clocks becomes a really central pillar of maintaining male health and vitality.
It's truly a profound and multifaceted connection, isn't it? Listening to all this, it really drives home how interconnected everything is. From the basic rhythms of our sleep-wake cycle to the intricate dance of hormones like testosterone, and even influencing our risk and treatment for serious diseases like prostate cancer. We've seen today how your body's internal timekeepers, these circadian clock genes, are so much more than just sleep regulators. They are intimately linked to testosterone synthesis, male reproductive health, and really our overall vitality. It reveals this powerful, often hidden operating system at work within us.
It really does. And as we connect this to the bigger picture, it's also important to, be realistic and acknowledge that while research is moving incredibly fast in this field, there are still exciting frontiers and unanswered questions ahead. For instance, that initial debate we mentioned about rhythmic clock gene expression and the whole testes versus specific cell types, like the Leydig cells, that really highlights the need for more advanced techniques. Things like single-cell sequencing or spatial transcriptomics, which let us look at gene activity cell by cell, or map it out in 3D within the tissue. That's how we'll truly understand the cellular diversity and precise timing within complex organs.
Right, getting a higher resolution picture.
Exactly. And while the animal models and the in vitro cell studies have given us robust mechanistic understanding, we still need more direct human mechanistic studies to confirm these pathways operate identically in people. And for chronotherapy, while the potential is huge for it to truly become mainstream clinical practice, we absolutely need more large-scale, multi-center clinical trials to really nail down the optimal timing for different drugs and different conditions and quantify the benefits robustly.
So, still work to be done, but the direction seems clear.
The direction is incredibly promising. But despite these ongoing questions, maybe this raises an important final thought for you, our listener, to consider. What if understanding and actively optimizing your own internal circadian clock, maybe through smart lifestyle adjustments like light management or meal timing, or perhaps through precisely timed therapies in the future, what if that becomes as fundamental as second nature to maintaining and improving male health as, say, diet and exercise are today? Imagine a future where personalized medicine routinely considers your unique biological rhythms for everything from balancing hormones to preventing disease. The potential for a healthier, more vital future simply by learning to align with our own amazing internal biology. Well, it's truly exciting to consider.